What we're going to try and do today is go through the content for Module 1 in biology, and we'll do it together as a group. And as we go through it, we'll do it as slowly as we can, but we've only got 2 hours. So if we feel we're okay with it, we'll move on. If we want to spend a little bit more time on it, we'll move on. So I'll go through a couple of slides. We'll stop and I'll say, okay, you can do the question at the bottom of that section on the Moodle, tick it off, and then I'll talk about the next page and so forth as we move through. By the end of the session, then you should have done first. So the cell function now is how do cells coordinate activities within their internal environment and the external environment. All right, so we're now going to look at how we link what comes into and out of cells back to that fluid mosaic model. Now, while in the syllabus it's a separate inquiry question, personally, I can't see how I cannot have fluid mosaic in with this bit, because this is fundamental to everything we're about to do. Now, when we do exocytosis and endocytosis and active and passive transport, it's all about this membrane. So it's going to be part of that dialogue, I think, now for quite a number of lessons. Some of it's new here and some of it's not. I can whiz through some of it quite quickly. Surface area to volume ratio. We've done that. That's fine. A few ideas to go a little bit deeper with that area. Concentration gradients is not new. But characteristics of the materials being exchanged is new. So, you know, what does this mean? What do we mean by characteristic? That's it. That's what you got. That's it. So I'll show you what I've prepared for you, and then we can discuss a bit. It's a bit like the table before, you know, 100 million examples. Well, let's choose the ones that are suitable to your school and to your students and your cohort. So modelling diffusion and osmosis, that's okay. Examining the roles of active transport, that's fine, we've done that before. But exocytosis and endocytosis are new. Then we go on and we look at all of this. When you teach it, is going to get mixed around, I think you'll find. I don't think you can teach it linearly. I think you'll find that you have to take a bit here, take a bit there, and move it together. So investigate cell requirements, and again there might be more, but we need to look at forms of energy, matter, gases, nutrients, ions, and removal of waste. And then we look at this one here, which is the interesting one because it says investigate the biochemical pathways of photosynthesis, cell respiration, and the removal of cell products and waste. So, yes, I'll show you what we've done, what I've done with that, and we can debate that, I think. Conduct a prac to, now this one's a bit different, conduct a practical to model the action of enzymes in cells. Now we've usually done create a model to demonstrate the action of enzymes. Well, that's this one, because it says investigate the effects of the environment on enzyme activity. And so that one would be your pH, temperature, substrate concentration, and enzyme concentration. That's the 4 of them instead of the 3. But this one is where we just created the model, the lock and key model or the induced fit model. Now it's saying here conduct a practical investigation. So remember the definition of practical. Now you know why it said field trip or doing an experiment in the lab. And I said let's add models to that. You remember when I said that right at the beginning? And I think this is why, because I can't think of another practical investigation you would do to model the action of enzymes. I don't know, unless you put milk on the, if you put some milk on the windowsill, and then you put some milk and added renin instead, it got faster. So therefore, is that modelling the action of enzymes? It's making it go faster. And I think maybe that you could, but personally I think that's going to be embedded with this.
One of the people out there in Adobe land has made the third comment, makes me wonder what the exam paper will be like.
Well, we'll find out when we get the first exam, so we'll be right then. That's okay.
I was at a consultation meeting on the new HSC exam specifications at the Board of Studies last week. They said at this stage there is no plan to release a sample HSC paper because under the new guidelines HSC has to be non-predictable, therefore they might not necessarily mean that the exam paper will have the same structure from one year to the next. So they're not locking themselves in by producing a sample HSC exam paper, but they will be, before the end of term 3 hopefully, producing a whole package of new descriptors, band descriptors, and they're going to try and address the injustice in science with the band descriptors being too high and so few kids getting the band 6 compared to other subjects, but they will also be putting out a whole range of annotated sample questions, right? So what the exam is going to look like is no one really knows at this stage.
And as to what's on it, we don't know that either, because I think what I've tried to do is cover all bases, but I bet I haven't. I bet there's still other areas that someone who writes the exam might do. That's their baby, so they're going to write a question about that, and we go, oh no, can't help that. So was there a question, Lorella? It's all right. The new textbooks. Yeah, I can't answer that. I saw a teacher gave me his review of the Pearson textbook, and there were quite a few gaps in the content compared to what's required here, but he's given them that advice. Now, if they fix that up, that's a good thing, but if they don't, well then that textbook's going to have a few gaps. And I suspect that will be the case across all of them. Yes. They're just, yeah. Yeah. They're racing to get the books out, so don't rely on any textbook at the moment. I'm just repeating what Alicia said. I think my advice would be you buy a textbook, every textbook, to check the stuff, what comes in. And you can buy one for me if you like. I'm happy to take one.
I jumped in last time the new physics syllabus came out, and the first book that came out was absolutely atrocious, bore no relevance to what the course was. So we are writing our programs in this program so that you do not need a textbook. You adopt the programs, should you wish, then you adapt it. Once you've seen the books that come out and you think one's good, go with that and modify your programs to suit. Well, yeah, of whichever one you think. But hopefully our programs will mean you do not have to buy a book to start with.
And we're trying to create resources along the way for you. And you guys are, like lots of people have created resources. So we might be quite okay once we come to term 4 and we have a look at the products, we might be fine. So there's a quiz at the bottom of that. Now I'm not going to go through those. Again, that's information for you to read at a later date, but you will need to know that information for the reason. This is on the Moodle, the fluid mosaic model. Oh, yes, I've gone back one, haven't I? We've started on cell function. All right. So once we finished inquiry question number 1, have you got that page up there on your screen? If we go back up here to cell SCI-BIO, top, we go back to our main page and we're moving on to how cells coordinate. And if you scroll down, you can now see what we're about to do in this section. I'll just give you a minute to find out where we're at. Oh no, that's the... So in the Moodle, now we've finished the first cell structure. So you might need to actually tick the box. You should have a box there somewhere to say that you've done it on the right hand side. No? So here, how cells coordinate is the second one. All right. So if we look at the first one, cell requirements for growth. Now I took that with a little bit of an interesting approach, perhaps a bit differently to what you might do. I talked about the first thing, you know, we need nutrients, we need water, we need energy, source of energy. And then I went, well, let's have a look at what do you do in the lab? What do you need to put into a medium to grow human cells in the lab? So if you were doing growing skin, what would you put in there so that the cells can grow? So I went, oh, I might look it up. And so I came up with this. I thought, oh, I could do something quite interesting with that type of information. So I've just listed it there for you, because it's full of words that you can take apart. I wouldn't teach it to them and say, you have to memorize all this, but it's a really interesting way to approach the requirements of cells, especially if you started with a context like, who's the scientist who grows skin cells? Fiona Wood, yeah. Oh, but for burns, that's right. It's about all cells. It's all about all cells, but you would start with this bit and say, well, in a human cell, this is the case. So do other cells have the same requirements? And then because this is all about microscopy, you could say, well, it's actually, that's a very good point. Because if you want to grow cells in the micro lab, you have to use a different medium because they need to supply different nutrients than you would if you were trying to grow a human cell. You know how we have red agars and black agars and green and blue and all sorts of different types of agar with the different nutrients to grow the different organism. Well, yeah, the different growth requirements, the different medium that's required to grow the pathogen. And then you have different temperatures, and there's a whole range of factors that are going to affect the growth. So it's a possible avenue to head down, which is very interesting. And the kids love to do agar plates, not that you do them here, but that's just a different take on that one. So then I created the characteristic of materials table for you. So you'll see that if you go on to the next page of cell function, characteristics of materials. And you can see it's quite substantial. And I thought, well, could do Fick's law if you wanted to do an extension there with maths and the equations. But for the general students, so I went through and I looked, okay, well, cells need oxygen. What is their characteristic? And I thought, well, okay, let's look at the size of the molecule, its polarity, is it dimolecular, is it linked to other molecules? That's a characteristic. Does it have a charge? So you've got ions down here as well. Then we needed to identify whether those substances were waste or whether they were needed by the cell. So I just went through and I put what's category and how does it actually move through the cell. Now, one of the reasons I put this last column here was about an opportunity to do problem solving here. If you look at the media, they are selling in face creams collagen. You know, you spend a lot of money on face creams with collagen. Now, collagen is definitely required by the cell. So it's a material that's required by the cell. But once you start investigating its properties, all of a sudden you realize that actually it's too large to absorb through the skin. Now, that's why something like this could be used as well, what else are we being sold that's a fake because of their properties? Yes, every ingredient has aqua as the major component. Yes. So again, this content is for you. Please do not give it to the kids in that structure. That's not its intent. It's just a learning for you to take on board, think about it and see if you wanted to add or change or reduce that information. Do you think there are any other characteristics that you might add to that? So I've done, characteristics. Insulin. Yes, I've got proteins. I've got proteins there. You could put example, I'll add insulin. Yeah, I'll add big, large macromolecules. See? Large macromolecules. Now that's a function of the molecule. You might do that as part of, transport. So you would use that as an example. Yeah, you would use that as an example to support what you're saying, definitely. Especially if you've got kids with diabetes. You do? We all do. It's terrible, isn't it? Any comment about any questions from the Adobe room, ever?
So the content that we've covered so far, just about cells, and we're probably not finished cells yet, are we? So how much time to do this in the syllabus?
So far, all of this is 29 periods. Take out the 8 periods for retreat, excursions, holidays, etc., maybe 21. But that's okay. Look at it. Remember, there seems a lot here. You're not going to get them to do that and read that and memorize it. That's not the intent. The intent is that they understand the ways things move into and out of cells, pretty much. And then what the membrane does in order to facilitate that. So if we move on, we've got 20 minutes left. So the next one is about concentration gradients. You can see there I've done no new, on the model there's no new information. That's exactly what we've done for many years. So nothing else new there. The next one is on diffusion and osmosis. Again, there's nothing new there because that's same old. Then we go on to active transport, which is not new. See, we're whizzing through this really fast. Then we get to endocytosis, exocytosis. It's really quite simple. We just have to remember that it's sort of here, it's known as bulk transport because the molecules are large. They're big and bulky. So that's why we use these processes of endocytosis and exocytosis there. Plenty of pracs around for that. That's not a very hard concept. I think you'll be fine with that one. There's plenty of resources on that page. Surface area to volume ratio is not new. That is in the current syllabus. But maybe you could expand it a little bit and look at the different shapes. So instead of just doing the cube, do some larger spheres, do some rectangular prisms, do some triangular prisms, and ask them to predict what they think will happen. Any shape that you can get the lab tech to create, really, because they're the ones who will have to do that. But that then, the minute you say, well, look, here's some different shapes. How? Yeah. Yes. Okay. That's me again. It must be getting time. Our heads are swimming, aren't they? Do you think you're getting a bit of an idea on this module, though? But it's not so bad, is it? Really, when you put all this together. So we can certainly talk about electromagnetic waves. Of course, that's going to cause a problem because they will have forgotten that. They will have forgotten the EMS spectrum. So you will probably have to go over that before you start. And look, the light spectra, the light absorption spectra, I think it's pretty important that they understand and can read that graph. I would never be expected to draw it, but I think to know that different wavelength is going to be absorbed in different chlorophyll, in different parts of the spectrum, I think that's important. So is that enough, do you think? There's not very much there, is there? Oh, look, I would never get them to do all of that. That's for you. I would only be going to this bit, the content. And I just draw a circle with a loop in it. That's it. Maybe, probably not. No, the fact that they use it, that it needs energy to happen. Yes. Oh, yes. Yes. The fact that the biggest one is that the light dependent breaks the water up and releases the oxygen, and that the hydrogen then is used in the light independent to create the glucose. Pretty much that's, to me, that's all you need. But you can certainly show them the picture, because the top chemist might be quite interested in that. And they might be appreciative of understanding that it's not just a simple direct reactant product, but there's a number of reactions. Oh, so YouTube's got lots of really good animations. Yeah. Okay. The extension, I thought maybe they might like to do CAN photosynthesis. That comes up when you do adaptations. So, you know, if you've got a really good kid, they might like to say, well, what happens to plants in the desert when they can't open their stomata and take in the carbon dioxide? What are they going to do? Well, then, there you go. There's some CAM photosynthesis. All right, so then respiration. Again, another 3 cycles. Kids will think they're really clever, won't they, if they can tell you the cycles that are involved in respiration photosynthesis. Again, we're not going to go into this in great detail, but that's just an overview summary. As long as they understand the products that are produced, and see, that's a nice example of Krebs. It's just to show what's coming out. Yep. Do they? At a nice simple level? Oh, does it? See, to me that's about all you need to show that one glucose gives 2 pyruvate, so that the Krebs goes around twice. So you get 36 ATP at the end. If you wanted to do Gibbs free energy, I'm not sure if that's in the chemistry. It is. So maybe it's a good point if you've got any chemists in there that they can look at that as a depth study, for example. So do they do that in year 11? In the first module? Well, we can say, in the last module of chemistry, you're going to be doing this in more detail. Now, for those kids who might ask you about anaerobic lactic acid fermentation instead of respiration, etc., I just put a few little notes there so that you've got the content there if you want it. I also added a little mind map that I drew, but that really similar. I did that from Moodle. So the information in the Moodle pretty much gave me the information I needed to do that myself. Yeah. So even if that's part of what the kids do, give them some text and ask them to convert it into, like, let them see something like that and then cover it over. So this is roughly where you're going to go. Now off you go and do it for me. Then they will put on it what they want to learn. And that might be the way to go. So the top kids can have more information and the others will have less. Then the last little part is about cell product and waste removal. I thought this was a really nice little image, and I says, give the students the image, but take out the sayings and ask them to fill it in themselves. I thought that was clever. The reason I put it here is because of the lysosomes, which is part of this one, and yeah, that's about it really. Vacuole. Oh, and the vacuole. So there's a structure in the vacuole called an acidocalcicones. I think I said that right. That's a new molecule that we've never done before, but I put it in that table because it's involved in vacuoles and the removal of and destruction of some substances that are going to be removed from cells, and especially contractile vacuoles. So it's in that earlier table. You might be able to use that if you're going to focus on that here, but that's up to you. So we've got the macromolecules, proteins, carbohydrates, lipids. They all have to move into and out of cells. We have wastes, which are carbon dioxide, toxins. So I talk about peroxisomes, the organelle peroxisome, what it's used for. Urea, and urea was in our table before as one of those materials or chemicals that move into or out of. Don't go into kidney function in any great detail here. It's just the fact that it exists and we have to get rid of it. Yes, no more aldosterone and ADH. No nephrons. I don't mind nephrons, but anyway. Now, I've really only given you a taster of what's in there, but I think you can see that that's quite a bit of content that should get you ready to face it early next year in with the program and without textbooks. So if you find anything that you think would be suitable to go in here, can you share it with me? If you find anything in here as you go reading it a bit later that you think shouldn't be there, let me know and I'll move it if you can give me a really good reason why. Or I can write, extension if you think it's too hard. Over the years, that's my plan, is that each year we evaluate what's here, add to it, and remove it if we think it's, or add extension to it if it's too hard or too much or whatever. Yes, I'm thinking this will be, so when a new teacher comes in, they've got the wisdom of the group all in one place. So you know how at the moment we find out new things incidentally in conversation, which is not a very ideal way to go about it. But my hope is that as the HSC exams come up, all those little tidbits go in here. All the little tricks that we learn from Meet the Markers about particular content areas that we need to question, they'll go in here. So the more you can feed to me, the more I can feed to everyone else. And then everyone benefits, sorry.
The interesting thing about this process that we're going through at present is we have PRIS and we have the Moodle. For you to get the hours for your QTC accreditation, for those people who are proficient teachers, you have to make sure that I have you enrolled in PRIS. Don't confuse PRIS with the Moodle. The Moodle will be out there and you can go into the Moodle as many times as you like. When will the Moodle really come into its own? I think the Moodle will really come into its own next year when people have got to teach it. But we've got the issue. We're going through these now. The reason we're going through these now is we believed that people needed to understand what's in course and the new stuff before we could finish the programs. We did a good job of starting the programs in term 1, and the graphs are pretty good. They're in a pretty good state, Now we're trying to refine people's knowledge and understanding of what the content of the course is. Then we go back in term 4 and get people to finish the programs. You then take them away after week 5 when they're supposed to be finished, adopt them and adapt them should you wish to do that. And then when you really get into the teaching next year, people, I can imagine, would go back to the Moodle because there's so much fabulous material in there to support you. But I don't know how, people don't do it. I think you were all probably enrolled in the Moodle, in the PRIS, and so once you finish all the exit activities, I'll transfer your name back to PRIS, and PRIS will give you your 5.5 hours for this Bio Moodle. For those people who've done the teaching stage 6 science for deep understanding, that carries 2.5 hours of NESA accreditation. So just, you might be able to help the people in your school understand that distinction between PRIS, that covers the hours. I can only tell PRIS you've done the course when you've done your exit tasks. All right? It's all very confusing. That's right, yeah. And I'm getting all these emails saying, I can't enroll in Moodle, and then you work it out, no, they're trying to get into Pris, you know, and I think, oh God, it's so confusing, but they're two distinct things.
Right. The reflective journal is a place for you to go and make notes for yourself or comments so that other people can go and read what you've said. Yes, so everyone will be going in there. So it's I think so. Have a look in there. Can you click on it now and show me what I think it can be commented on? Well, we can read it, yes, but it was on the very first page. So if you go to bio at the top it always shows you when we're discussing where the reflective journal is and what's required for it. A comment somewhere throughout the Moodle about something and comment on two others. Give feedback to two others in your cluster. So this is where the clusters will come up. So you'll only see the people in your cluster. And so you post a comment and say something about, I think the Krebs cycle only needs to be done at a very basic level, and I'm not going to do the chemicals that are created through the Krebs cycle because the kids don't need it. However, I might give it to those kids who are doing chemistry because of such and such a reason. Something like that. Yep. Don't worry about the journal. That's for you. Just make a comment. I can always delete that. I think you've probably got enough to do. Thanks everyone at home. Our heads are exploding here, but I do hope that gives you a little bit more confidence about ready to start in term 1 next year. All right, thanks everyone at home. Bye for now.